Hello Vanessa and hello everybody. I'm Emilie Schol, Business Development Manager at Life Sciences, and I'm specialized in biotech and pharma. And today we are going to present you range assays dedicated to drug discovery and drug development in the field of immunology.
So in this webinar, we will try to follow the different steps involved in the immune response using the example of an infection occurring after a skin injury. We will present the test we provide to mimic these mechanisms and, of course, to evaluate the potential of your compounds to influence these processes. We took the example of skin, but in the next slide, we will also present the other applications that can be relevant with our assays. So the population of immune cells is quite large and heterogeneous. So in this presentation, we will only focus on the main cell types and mechanisms involved in both the innate immune response, which is the first and aspecific response, and the adaptive immune response, which is much more longer and specific. So one of the first process engaged is called phagocytosis. Specific cells such as macrophages, dendritic cells, and neutrophils, PMNs, ingest and destroy without any specificity foreign organisms such as bacteria or viruses. So we have developed a test to mimic this phagocytosis process. We take PMN, we leave for a short incubation period, 45 minutes, with fluorescent beads. PMN internalize those beads by phagocytosis and then become fluorescent. We can then analyze and count fluorescent PMN by flow cytometry. Here are some examples. So here in the background control, we do not see any fluorescent cells. And after the addition of beads, we can see the phagocytic PMN that become fluorescent, indicated that basal phagocytic activity of the cells has occurred. And by adding a stimulating reference, here FMLP, we observe an increase in phagocytosis. Indeed, we have much more cells in the top of the plot. So an increase in fluorescence intensity indicates an increase in phagocytosis. So by adding your compound of interest during this process, we can easily observe if they can facilitate or not the phagocytosis process. This assay allows the testing of compounds that may stimulate the immune system or increase defense against pathogens, and it may give information on, for example, potential bactericidal activity. Furthermore, it has been shown that an accumulation of debris that has not been internalized can disrupt tissue homeostasis by exhibiting immediate cytotoxicity on cells and can induce chronic inflammation leading to tissue damage. So by phagocyting aged cells or cellular debris, PMNs can also play a role in tissue remodeling and regeneration during aging and wound healing. Beside their primary function of phagocyte of the innate immune system, there is an increasing evidence that PMN may also play an important regulatory role as mediators of inflammation via the production of ROS, reactive oxygen species, and cytokines in response to infection.